Right, will you be able to see this?
Yes, doctor.
Yes, doctor.
Okay,
Yes.
good. So today we're going to talk about media,
Doctor.
right?
Recording in progress.
Okay, so good afternoon, everyone. This week, we're going to talk about media. So before we get started, do you have any idea what media is? Any idea? Why do we need media for the cell culture?
For them to grow.
Okay, make them to grow. Okay, you got to remember that because you excise, you get the cell from the part of the animal, from the animal bodies, okay? You excise it, and then you try to grow that in an artificial media, all right? To make sure that the cells are alive, that they can survive and they can proliferate. So remember that media is the utmost important medium for the cell to grow, okay?
Yes.
Okay, so nutrient mixture is the cornerstone of the cell culture because it provides essential nutrients that are incorporated into the dividing cells. So having the correct having the correct nutrients mixture can often be the determining factors in failure or success in growing a cell in vitro. So always remember that. Okay, you have to be very careful with your media when you culture your cells, and you must know your characteristic of your cell line. Okay? Right? Because it is very important for you to understand, to know your cell line before when you do the cell culture, so that you can choose the correct medium or the correct nutrients or media for them to grow. Right? So they provide very essential nutrients incorporated into the dividing cells, such as amino acid, the fatty acid, okay, the sugar or the glucose, ions, trace elements, vitamins, cofactors, and other ions and molecules, which is necessary to maintain the proper chemical environment for the cell. And you also got to remember that each cell, they need different nutrients, because they have different character. Some may need high sugar, some may need low sugar, some may need extra essential amino acid, some probably need different kinds of vitamins and also fatty acid. So you got to characterize, you need to know well the characteristic of the cell when you choose your media, okay? So it is best to initially grow the cells in their original medium and compare this with the more convenient medium after a passage or two in each. Passage means you do subculturing. But what happens if you do not have any idea on what types of medium the cell could be grown? Okay, because you don't know the original medium. What you need to do? So the growth rate and morphology of the cell look the same, then a medium switch can be. If you have the original medium and you try to, adjust it so that the morphology of the cell will look alike, then you can switch your media. Okay, you can switch your media to the one that you adjusted. Okay. And the medium contains all the parts of the buffering system required to maintain a physiological pH and should provide the appropriate osmolarity of the cell. So these parameters we want to talk about in the coming lecture, all right, which is the physical environment and also physiological environment for the cell to grow. So that one we're going to talk about in the next lecture. But for this lecture, we're going to focus on the medium itself, right? Is that clear? Is that, can you follow me?
Yes. Yes, doctor.
Okay, good. So keep in mind when trying to repeat published data, that's the cell growth in the different medium, they may respond differently in the same parameters measured. Or sometimes even though you follow the parameters in the published paper, It is not necessary that you can replicate the result. You got to remember that the chemical that you use, the brand of the chemical that you use, the batch of the chemical that's manufactured could be slightly different. And also the physical environment in our country and in the other countries like in the US or in the Europe, they could be different. And they also, this come into our considerations when you try to optimize the proper parameters for the cell to grow. Okay, so you don't actually blame that you cannot replicate, because sometimes these factors have to take into account when you grow your cell line. And it is best to screen several of the commercially available media if no published data exists. Okay, sometimes you don't have the published data. So what you do, you probably need to choose a few different commercially available media, and then you grow the cell, and then you choose which media give the best result, and you choose that media. All right? Does that make sense?
Yes, doctor.
Okay, good. So compare the formulations of the media tested and look for patterns of whether the cell of interest seem to prefer a low or high glucose, low or high calcium, low or high level of amino acid, and so forth. So always remember, as I told you just now, each cell, they have their characteristic and they have their own liking. Yeah. So you need to understand the characteristic of cell line so that you can optimize the media so that the cell will grow in the more conducive environment. So it is useful if you decide to further modify the media. Okay, especially come to modifications of the media. So you need to look at the characteristic of a cell and also the morphology of the cell after you grow them on the media that you adjusted. Okay. So the media can be purchased as prepared liquid media. Of course, this one is more costly because you just need to use it straight away. You don't have to formula them, or you don't have to mix them and then you autoclave them. For the ready-made or the ready-made liquid media, you can use it directly. Or you probably need to just heat it up to the room to the temperature that you require, and then you can just straight away transfer the cell onto the media. Okay, depending on the types of cell line that you use. Or you make up in the laboratory from a dry powder mixture. You also can make up your own media by mixing the powder that are available in the lab, or prepare in the laboratory from the individual stocks of the individual components or groups of the component. Or you can prepare your own from the individual stock that is in the lab, which is available in the lab. Okay, so there are probably three types of three kinds of methods that you could choose to generate your own media. Okay? Boleh ya? Boleh. Can you follow?
Yes. Boleh, doctor.
Okay. Now, media components deteriorate with time, so do so faster in solutions. If they are in the dry powder form, they probably not deteriorate as fast as when they are in the solution. So always remember that. Okay, some necessary components break down and are lost. Other correct toxic breakdown products are oxidized to toxic components. So it is always safe to store the pipette the powdered media and make liquid media in the laboratory on a regular basis. For instance, yeah, when you plan your experiment, you have to be very careful. Okay, when you wanted to culture your cell, okay, in next 6 months, you don't prepare the media now because the duration is too long and the nutrients could have been break down, and the properties of the media may have lost its own properties. Okay, so it is best you prepare the media freshly. For example, you want to culture the cell in next 2 days, so you can prepare it today or tomorrow. So the media always fresh, and you have all the necessary components freshly, which is for the cells to grow. Okay, make sense?
Makes sense, doctor.
Okay, now, so the powdered media and nutrients mixture generally have a shelf life of a year or more if you store in a moisture-proof A-type container in the dark. Always remember that some of the chemicals, they are sensitive to light. So it is good if you can store them in the dark. You wrap it with the aluminum foil, okay, and with the A-type container, and you can so that you can prolong the shelf life of the chemicals, okay, or the nutrients. Or you're preparing the media in the laboratory from a component immediately before use. That's the best, which is obviously the best way to ensure that the media contain the desired components in the desired form. And also it is essential if the investigator wishes to study the role of the nutrients themselves and to optimize the nutrient portions of the media. So for example, if you wanted to understand the media, the effect of the media on the growth of the cell, okay, you probably have different media. So you need to make sure that you prepare the media at the same time. You don't prepare them one media which is a year ago, or maybe one media 6 months ago, and the other media which is you prepare it now, and then you compare the effect. That cannot be compared because the durations is different. The times that you prepare the media is different. Once it's already prepared years ago, 1 year you prepare 6 months ago. Okay, so the properties of the media already change and the parameters also change. So you cannot compare them, especially if you wanted to understand the role of the media on the growth of the cell. Okay, so the preparations of the mediums from the commercially powdered nutrients mixture and a limited storage of the prepared media in a light type refrigerator will be adequate. So it is less costly than purchasing prepared media, especially when the cost of the filter, so forth, can spread over the large volume use. So sometimes, remember, you need to filter your media. Okay, and the filter can be quite expensive as well. So you need to always see the budgeting. You always need to do the good calculations. Which one is better? Which one is more economical? Whether you prepare it from the powdered nutrients, or you from the powdered nutrients mixture, or you purchase it directly from the manufacturers. So you always need to do the comparison of the cost before you proceed with your experiment. Okay, so as a young scientist, or as a lab manager, or the project leader, or the students, you always need to be careful with your costing. Okay, because you need to make sure that the cost that you use is in the most economical way. All right. So what other components are found in the complete media? What can you find in the complete media? First, you will have the amino acid, the vitamins, salt, the glucose, the fatty acid, the organic supplements, hormones and growth factors, and also antibiotics. And each of these components have its own role. Okay, when you culture your cell line, right? Now we look at one by one. We look at amino acids. So amino acids, the essential amino acids, which are not or cannot be synthesized in the body together with the cysteine and tyrosine, generally are required by cell culture, the cultured cell. Okay, remember that cysteine and tyrosine cannot be generated by our body, so you have to get it from, you have to outsource them. Okay, so other non-essential amino acids are often added, okay, to compensate for the particular cell types and capacity to make them, and also the glutamine. Okay, the glutamines required by more cell, and for some cell lines, they use glutamine as a source of energy and also carbon source. Okay, so always remember that. Okay, the requirements, the characteristic of the cell is different. Okay, for each cell line, and the amino acid required also different. All right. Okay.
Okay.
And if you look at the vitamins, there are so many types of vitamins that are available, and many media contain the common vitamins such as niacin, folic acid, riboflavins, cetol, thiamines, and so forth. And this essential is very essential to make sure that this continuous cell can be replicated. It's very important for cell replications, and it can be a detrimental factor or effect which is might not seen until several cell doubling after their removal from the media. For example, if you remove niacin or you remove folic acid and inositol, so after a few cell doubling time, then you can see the effect. All right, probably at the very beginning when you remove this specific vitamin, you might not see notice the effect, but after few cell doubling, then you can see the effect. Okay, probably the cell doubling times or cell replications is getting slow because of the absence of this specific vitamin. Okay, and apart from that, you also have biotins, the PABA, which is the p-aminobenzoic acid or fat soluble vitamins such as ADEK. Okay, so that's why you need fatty acid in your media. Okay, so because some of the amino acids, which is, for example, the ADEK is fat soluble. They only can be absorbed by the cell when they are carried by the fat. Remember that? Okay. And this is types of the salt. Okay, the salt that are available. You know, there are many actually. The components such as the calcium chloride, the kalium chloride, the kalium sulfate, magnesium chlorides, okay? And in each of these brands, for example, the OBPS, the Dubilco PBS, okay, but Lee, Hanks, and Spanish salt. This, you see, each of the contents of the salt is different for different brands. Yeah, because the cell preference towards the salt solutions also different based on the characteristic. Okay? Some salt, they probably need more calcium ion. Some probably need more magnesium ions. So they are different, right? Okay, so they provide ions, okay? Whether it is an ion, a cation, for example, the natrium ions, the kalium, or magnesium. Magnesium should be 2+, huh? There's a typo there, the calcium, the chloride, the sulfate, or phosphate. And this is important to regulate the membrane potential. Do you remember that the calcium pump in the membrane bilayer?
Yes, doctor.
Okay, so there's a channel. Okay, when the calcium is in flux, then the kalium will be channeled out. They're in and out, and there's some membrane potential, and although the voltage change on the membrane potential, which we're going to discuss in more detail in the coming semester in more physiology. All right, and the matrix requirements are nutritional closure for the macromolecule and also regulators for the intracellular changes such as the sulfate, the carbonate, and also the phosphate. Okay, so in suspension culture, less calcium is needed to reduce the cell aggregations, because sometimes in the suspension culture, the cell may clump together if you have too high calcium. So you need to reduce that so that the cell will not be aggregated. Okay, and also this the sodium bicarbonate concentrations is also determined by the carbon dioxide in the gas phases. Okay, in addition to the bicarbonate, the carbon dioxide buffering system, the media may also contain some buffer and perhaps in a complex organic buffer, because you need buffer system to make sure that the pH can be maintained at the desired level. Okay. And glucose, of course, is very, very important, because glucose is the food for the animal cell. Okay, especially for the carbon source, the source of energy. So they metabolize the principle by the glycolysis to form the pyruvate, which which will be converted into the lactate and also the acetolactate and enter the citric cycle to form the carbon dioxide. Remember that the glucose, which is C6H12 and O6, which when they are being hydrolyzed, they will become what? No idea. What happens when glucose is being hydrolyzed?
Carbon dioxide.
Carbon dioxide and
Water.
water. Okay, that's very good. Okay, perfect. Okay, so glucose will be, okay, after being hydrolyzed, they will become water and carbon dioxide. Okay, so media also contain lipids. Most contains a mixture of the fatty acids, and some contain more complex lipids such as cholesterol. So in some media formulations, such as medium 199, they contain detergents, for example, twin 80, to help to emulsify the lipid. Okay, and these detergents can be toxic to some cell types, so particularly in the serum-free media. So when you use twin 80, you also need to be very careful, okay, because sometimes they can cause toxic to the cell. Okay, but they're useful to emulsify the lipid. Okay, for organic supplement, which may include proteins, the peptides, the glucoside, pyruvates, lipid, and citric acid cycle determined intermediates. This can be supplied directly onto the media. Okay, and also the albumin, the globulins, fibronectins. Okay, fibronectins are used to promote cell growth. The alpha microglobulins to inhibit the trypsin, and ferritin, the laminins, which enhance the cell attachments, and also the transferrins, which is very useful to bind the ions and make it less toxic. Okay, these are the proteins also added to the media. Okay, for the cell survival. Okay, and apart from that, we also have hormones and also growth factors such as insulin. Okay, insulin, as all we know, is promote the glucose uptake by changing the glucose into glycogens, and may also have the effect on the mitogenic activity via the IGF growth factor 1, and also apart from that, the growth factor also involve the insulin growth factor 1 and insulin growth factor 2, and also the platelet-derived or PDGF or platelet-derived growth factors, which can stimulate the growth in the fibroblast and also the glial cell, and also the fibroblast growth factor, the FGF, and also epithelial growth factor, the EGF. Okay, these are the growth factors which is necessary to be incorporated into the media. Okay, for some cell type, for certain cell type. And also antibiotics also will be added into the media to reduce the frequency of contaminants, because the major problems in animal tissue culture is the contaminations. So once you're cell contaminated, all the data will be gone and you cannot collect any data. Okay, so the utmost important thing is to reduce the contaminations. Okay, the use of laminar flow hood coupled with strict aseptic techniques make antibiotic unnecessary. If you have very good aseptic technique, if you have very good laminar flow hood, then probably you can omit the choice of using antibiotics, because too much antibiotic being used also not good for the cell. Okay, because you make the immune system of the cell become less, and also you make the immune system of the contaminants become stronger. Okay, so sometimes you probably need higher dose of antibiotic just to kill the kill the contaminants. Okay, these are the commonly used antibiotics in tissue culture. Okay, such as our amphotericin B, which is also commonly known as Fugizont, which is very useful to treat fungi and yeast infection. The working solutions is at 2.5 microgram over liters, and if you use too much, which is exceed 30 microgram over liter, then you know they will cause toxicity to the cell. Okay, antibiotic is good, but too much, they'll cause toxic to the cell. And apart from that, we also have gentamicin and clindamycin, which are also streptomycin, which is very useful to treat gram-positive and gram-negative bacteria. And nystatin, also very good to treat fungi and also yeast. And of course, we have penicillin, which is very good to eliminate the gram-positive bacteria. So this common antibiotic you need to remember, okay, the functions of this antibiotic. Okay, all right. Boleh ya?
Boleh, doktor.
Okay, good. Let me see, there's a chat box there. Somebody say what? Huh? I can see. Okay. Like, what is the disadvantages of using antibiotic? Of course, you encourage the development of the antibiotic resistant organism. Okay, so we don't want this to happen. Okay, that's why if you misuse antibiotics, you can cause a very serious problems of encouraging the existence of the antibiotic resistant organism, okay, or what we call the superbugs litter. It is recommended that the use be restricted to the primary culture in large scale level. Okay, and pH, this is also one of the very important parameters, and phenol red is often used to as a pH indicator. Okay, you can see there's a pinkish color. So if the pH drop, then the color of this phenol red will change to orange or yellow. Okay, so it's a rapid types of assessing the pH of the media of all the culture, and phenol red can be added to the media if it is not part of the media powder, or if a more obvious colors is desired. So it has a weak estrogenic activities, which may be a concentration in some cell. So you need to be careful when you come into the use of the phenol red, the amount of the phenol red used in the cell culture. Okay. So serum also very important, and calf serum is all widely used together with the fetal bovine, the human, and also the horse serums. Okay, because they may contain the protein, polypeptide, hormones, nutrients, and also other metabolites and minerals, and also inhibitors, which is necessary for the growth of the cell. Okay, but we also have the serum-free media, which is available because some of the cells, they prefer serum-free media. Okay, culture may be maintained and proliferate without serum, right? For example, the L929 and HeLa cell line were among the first to be grown in the serum-free media. Okay, L929 and also the HeLa is the cell type or the cell line generated which do not need serum in their media. Okay, and these are the advantages of the serum-free media. You can just read on the common sense, and you also have the disadvantages of the serum-free media. All right, so these are also the common sense. So you can just read and you will be able to understand. Okay, and most media, such as the minimal essential, we call it ME, or the Bell modified eagle, the DME media were developed specifically to use with the serum supplementations and high density of the cell growth. Okay, others such as the HEMS nutrients mixture, the F12, F10, and also the molecular cellular development biology, the DM series of the media were tailored specifically for growing a given cell types at low density with a minimal amount of undefined protein added. Because some of the cells, they can grow in high cell density and some they cannot. So they need a specific formulated media for them to grow. Okay, for example, the F12, the DCME media work very well for growing cell at low or high density in defined conditions or with serum. Where media for growing mammalian cell have similar components, while the insect cell culture, for example, the breast media is quite different, reflecting the different metabolic needs of the insect cell. So remember that, as I said earlier on, the characteristic of the cell lines is different. For example, the mammalian cells and also the insect cell, they have different characteristics, and hence, they also have the different metabolic needs. And that's why they also need different types of media for them to grow. So it makes sense, right? Logic?
Yes, doctor.
Okay. So the other types of the media, like Lebowitz L15 media, is desired to grow in the cells in equilibrium with the A rather than the CO2. It's useful when CO2 incubator are not available, especially like in teaching laboratory. Because if you have the CO2 incubators, it's quite expensive, yeah? So when the cell achieve or the latent intensity outside of the incubator during the long tissue association protocol, so this media is good for that particular types of cell to be, okay? If you do not have the CO2 incubators, okay? So preparing media is easier. For example, the region given for preparing 1 liter of media. So for example, you have the DME powder. These are the types of the media that you prepare in the preparation room. Remember that you have a preparation room in your lab. Can you look at the floor plan in a previous class? So when you prepare the media in the preparation room, right? So this is the protocols. So when you prepare 1 liters of the media, you don't straight away put 1000 ml straight away and then mix everything in. That's very wrong. Okay, because the end volume will be much larger than 1000. So usually you put 750 or 800 ml. Okay, then you mix them. At the end, you top up with the extra solution so that the end volume will be 1000 ml. Make sense?
Yeah.
Do you know,
Yes.
have you prepared any media before or solutions before? Or have you been taught to prepare solution before?
Yes.
No. Okay,
Yes.
good. Okay. And testing media and the component quality. So you always need to keep one set of the glassware, which is exclusively for the media making. And you need to rinse well with distilled water, but not wash with the detergent, because detergents can stick on the wall of the glassware, and they can be incorporated accidentally into the media and cause very severe effect for the cell. And the sodium bicarbonate and other reagents that are used in the media should also be used only for the media. You don't mix it to prepare other solutions. Okay, so when weighing out these reagents, a disposable disperser or the wet box should be used. Okay, this is to avoid the contaminating this reagent with others. So potentially a toxic chemical that may be used in the laboratory. And if you don't change your weighing board, okay, so you know this contaminant may get into your media and to be toxic to the cell. Okay, and water purity also very important for good quality of the media. So you don't simply use tap water to mix your media. Okay, and some cell type can be extremely sensitive to the poor media quality even when serum is used. So the lot test media powder, the critical hormones, and also the serum are by enough of the best lots to last for a year. Okay, so lot testing would include the testing of the previously used lots of the media, for example, against 2 or more new lots. Okay, this is very important to make sure that the media and the component can be controlled in terms of the quality. Okay, at the same, all 3 batches of the media should also be made out on the same day, with only the different being the media, such as the same water and sodium bicarbonate and other and etc. And the testing should include daily growth curve from which one can calculate the lag, phase and population doubling time, the exponential phase and saturation, the saturation density. This will be discussed more in detail when we come to the coming lecture later. So it is also a good idea to do a plating efficiency experiments. Okay, we also will talk about plating efficiency in the coming lecture, since cells at low density are more sensitive to toxic components, while high density growth would be better to detect the media deficiency in the nutrients, because higher the density of the cell, the medium will be exhausted easily. So it's easier to know to understand when the nutrient become deficient. For the troubleshooting, this is also common sense. So as a student, or okay, maybe in the future become a lab manager or whatever, the PI. So you need to understand how to troubleshoot. You'll be the investigator if sometimes thing may happen unexpectedly. Okay, you probably use the media like last month. You did it fine, everything is fine. But when somebody use it for their experiment using the same media, but something worse happened, probably the cell line just gone like that, or they cannot grow it. So you need to troubleshoot the media. What's happening? What gone wrong at some point? So you need to investigate that. Okay, these are the tips that you can refer to when you come to troubleshoot your media problems. Okay, because it's happened quite frequently for media problems. Okay, so knowing the troubleshooting is very important so that you can rectify the problem in a much faster way. Okay, so these are the further readings that you may read. Okay, and that's for now. Okay, any questions you need to ask?
Recording stopped.
Any question, class?
Doctor,
Yeah.
as you mentioned before, what is lot test?
Oh, the lot. Okay, remember, each serum, when you purchase, they have different lot, right? Or the chemical that you purchase probably have different lot or different batch, right?
Like different component like that?
No. Lot ialah the batch they produce. Bila dia produce the lot. So when you produce something, you have different lot kan?
The version?
Yeah. So
Okay.
contohnya masa dia produce mungkin this month, last month, previous month. So there is different lot kan?
Yeah.
So when you buy something, they also have some lot number contohnya. So kita kena tahu ya, sometimes this lot may not come to the quality that you expect. So kita kena selalu buat lot test ataupun media yang kamu sediakan tu, dia ada lot media dia sendiri. Ada yang media yang kamu sediakan pada minggu lepas, ada yang kamu sediakan pada minggu ini. Ini jadi dua lot dah, because you sediakan dia pada masa yang different. Boleh faham?
Okay, thank you, Doctor.
All right. Any more question? Ada yang perlukan attendance? Ada tak?
Saya, Doktor.
Kamu group mana? Group 2 kah? Group 3?
2.
Sekejap saya share attendance nanti. Sekejap.